well, good morning. I'm Villanueva-Alvaro, and I'm doing my PhD at the University of Basque country, and I'm going to talk about or studying magnetic hyperthermia of Magnetovibrio blakemorei. Okay, well, first of all, Magnetovibrio blakemorei is a magnetotactic bacterium. These are microorganisms with the ability to biomineralize magnetic nanoparticles called magnetosomes, surrounded by a lipid bilayer membranes. As you they organize magnetic nanoparticles forming chains along their longitudinal axis, as you can see here on this time image. And they synthesize these nanoparticles to align and navigate along the geomagnetic field lines to reach optimal regions to live. Okay, well, in this work, I'm going to focus on Magnetovibrio blakemorei. This magnetotactic bacterium have magnetosomes with magnetite compounds and a truncated hexaoctahedral shape, as you can see here on this TEM image and in this schematic representation. It's important to note that the morphology, size, and distribution of the magnetosomes depends on the magnetotactic bacteria species. Here you can see some magnetosomes with different morphology, for example. Okay, and why do we work with magnetotactic bacteria? Well, magnetotactic bacteria have promising properties. Here you can see some of them. For example, they can swim by navigate by their own. They can respond to external magnetic fields, and also they can detect regions with different compositions, having a special preference in tumoral tissues. These properties explain why magnetotactic bacteria are widely studied as a promising nanobiorobot for biomedical applications. Applications like, for example, external navigation control for drug loading, drug release, transport cargo, and even to apply localized therapies like magnetic hyperthermia. In this work, I'm going to focus on magnetic hyperthermia therapy, especially in the heating efficiency of Magnetovibrio blakemorei. And I'm going to show you some experimental and simulation results, and I'm going to compare the heating efficiency of Magnetovibrio blakemorei with the heating efficiency of other magnetotactic bacteria. Okay, well, first of all, the term magnetic hyperthermia refers to a treatment where there is a temperature increment produced by the action of an AC magnetic field. This treatment can be used to fight against cancer, for example. And I'm going to explain that using these images. In a magnetic hyperthermia treatment, the heat source is introduced inside the body in the tumor area. This heat source, it can be, for example, magnetotactic bacteria. Once located in the tumor, the heat source increases the temperature in the area due to an action of an AC magnetic field. It's well known that increased the temperature between 41 and 44 degrees leads to the death of cancer cells without affecting the healthy ones in the process. So this explains why magnetic hyperthermia is a powerful tool to fight against cancer. But if we are going to use magnetotactic bacteria as a heating source, we need to know their heating efficiency. And the parameter that describe that is the well-known as a specific absorption rate, or also called SAR. As you can see here, SAR is defined as the absorbed power normalized by the mass of nanoparticles, the mass of the heating source. Okay, well, the heating efficiency can be measured by two methods, calorimetry and magnetometry. The first one needs the measurements of the temperature increment of nanoparticles under an AC magnetic field. But the main problem with this method is it requires adiabatic conditions. On the other hand, magnetometry method needs the measurement of the dynamic magnetization. And as you can see here, SAR is directly related to the integral of the AC hysteresis loops, which correspond actually with the dissipated thermal energy. Okay, on the other hand, as you can see here, that we have carried out some calorimetric measurements using a sample of Magnetovibrio blakemorei with this mass of magnetite and this volume under this frequency, but under non-adiabatic conditions. But later I'm going to show you how an adiabatic corrections can be made to these experimental results in order to estimate the SAR values. In addition, you can see here that we have carried out also to magnetometry measurements And we will use this AC hysteresis loop to calculate the SAR. And then later I'm going to compare both results that we get with calorimetry and magnetometry. Okay. Well, now I'm going to show you how we have done the adiabatic correction to calorimetry measurements. First of all, here you can see the experimental results before and after performing the adiabatic correction. You can see also the differential equation governing this case. Under adiabatic conditions, there is no heat exchange with the surroundings. So the temperature variation with the time only depends on the heating source. However, under non-adiabatic conditions, there is a factor associated with the heat transfer losses. So the adiabatic correction consists only of adding this term to the experimental results. To develop that, we need two parameters, room temperature and tau parameter. First, I obtained the tau value by fitting the experimental results to an exponential function. Is the solution of our differential equation model. Okay, so we add this heat loss term to the derivative of the experimental data, and then we integrate, and we obtain the results under adiabatic conditions, experimental results under adiabatic conditions. So we use these corrected results to estimate the SAR values. And as you can see here, the maximum SAR values that we get is around 400 watts per gram. And as we expected, the SAR values increase with the applied magnetic field, as you can see here. Okay, on the other hand, we obtain the SAR values using the measured AC hysteresis loop of Magnetovibrio blakemorei. Okay, as you can see here, the heating efficiency is null below 40 millitesla, and the SAR has a growth curve until it reaches the saturation around 80 millitesla, more or less. And the maximum SAR value for a frequency of 132 kilohertz is around 2,200 watts per gram, as you can see here. And to compare magnetometry results and calorimetry results, we need to normalize the SAR values by the frequency, because SAR is linearly dependent with the frequency. And if we do that, as you can see here, the results obtained by calorimetry and magnetometry are compatible. So that means that we have done a well-corrected adiabatic correction. Okay, well, on the other hand, to understand the physics behind the experimental results, we have simulated the heat transfer of a Magnetovibrio blakemorei samples. For the simulation, we have used console multiphysics and with many experimental parameters, as you can see here: materials, geometry, heat power source, emissivity, and so on. We have also considered the heat transfer phenomena, all heat transfer phenomena, conductive, convection, and radiation. Radiation is important to the heat transfer process in order to get the compatibility between experimental and simulation results. Well, after taking all these experimental parameters into account, we meshed and we solved the problem, and we get, as you can see here, the experimental and simulation results for both magnetic fields are compatible. So we were able to simulate experimental measurements by calorimetry. And the next step, we will use this heat transfer model to simulate the heat transfer in biological systems. For example, biological systems like 3D tumor spheroids, for example. Okay, finally, you can see here these are values of 3 magnetotactic bacteria. Okay, Magnetovibrio blakemorei, Magnetospirillum magneticum, and Magnetospirillum griffithswaldensi. Okay. And as you can see here, Magnetovibrio blakemorei has the highest SAR saturation value, around 2,200 watts per gram, which is around, around 2,200. The reason for the difference in SAR values is due to the morphology and organization of magnetosomes. As you can see here on this slide, Magnetospirillum magneticum has a fragmented chains of magnetosomes with a slightly deformed truncated octahedral shape. And Magnetospirillum griffithswaldensi has only one chain of magnetosomes with a slightly deformed truncated octahedral shape too. So according to these SAR results, it's possible to select the appropriate species according to the specific biomedical applications. So, in summary, as you can see here, we have seen the effectiveness of the adiabatic correction. We get compatibility results between magnetometry and calorimetry. And also Magnetovibrio blakemorei have the highest SAR saturation value than other magnetotactive bacteria species. But the problem with that is require a higher magnetic field to reach that SAR values. And also it's possible to select the appropriate species according to the biomedical applications that we want to do. And finally, finite element simulation can be used to evaluate the heat transfer in biological tissues. And that's all. If you have any questions, thanks for your attention.
Thank you very much for a very interesting presentation, Danny. The floor is open for questions. Who would like to ask a question, please raise a hand or text the question in the chat. Okay, if no one's asking question yet, I will ask one. So recently I've heard on one conference that for the correct estimation of SAR values, some groups not only use the heating curve, but also the cooling curve. Have you tried something in this direction as well? Danny, you're muted. Could you please unmute yourself?
Sorry, can you repeat the question, please?
Sure.
It's about
Sure. Yeah. I was asking whether you can also, or you try to also use the cooling curve for the correct estimation of SAR value. I just recently heard on the conference that some groups actually try to do that, and they claim that it leads to more precise results. That's why I was asking.
Yes, it's possible to use both heating curve and cooling curve, and you can get using this differential equation and this exponential function, well, with some little change, you can use it and you can get a good value of the SAR values.
Okay, but you only use the heating curve.
Yes, in this case, yes, but we tried with other magnetotactive bacteria samples, and we can use the cooling curve too, and we get the same results. So for that reason, we only use the heating curve because it's not necessary for us using the cooling curve too.
Thank you very much for the explanation.